Thank you for a very kind introduction, and it's my great pleasure to attend the TAMEST conference and share with you our recent work. So let's see how it's going to work. So through our life, if we were lucky, we could be immune from any diseases. Actually, this is almost how my grandma spent her 100 years life, so which is wonderful. But aging is different. So far, we don't have a way to avoid it. So it's happening to all of us. And aging not just change how we look, giving more wrinkles and less hair. It is a progressive process with gradual decline in organism fitness. So during aging, different organs in our body undergo fundamental changes. And many of those changes are associated with increased risk of different chronic and degenerative diseases. So as a result, if we look at senior people over 65 years old, compared to younger population, the prevalence of most chronic conditions increased dramatically, as shown in this chart. So one exception here is depression. So we are happier when we are getting older, which could be a cheerful thing to look forward to. But for other age-onset diseases, such as cardiovascular disorders, cancer, and Alzheimer's, the likelihood to have those diseases are much higher in elderly people. And treating those diseases are generating a huge burden to public health in our current society. And this situation won't become any better in the next few decades with world population age. So it's estimated between 2014 and 2050, the percentage of the population at age 60 and above will be doubled from 11 to 22% worldwide. And in some countries like Canada, most European countries, and China, where I come from, this number will be even higher, up to 1/3 of the population. So with this dramatic increase, how can we keep our senior population in a healthy condition is a big challenge. And for myself, as a basic biologist, to tackle this challenge, I think we need to better understand what are the factors that can influence the aging process, and how can we use them, or manipulate them to promote healthy aging. So first, let's look at what are the factors involved. So this world map shows you 5 longevity hotspots where people have exceptionally longer and healthier life. One example is in Korea, in Greece. There, in every 3 people, 1 can live their 90s. And this population has almost no dementia. The other example is in Sardinia, in Italy. So there, they have the highest percentage of male centenarian. Unfortunately, compared to females, male centenarian are quite rare in other places. So it's amazing that people living in those areas have this longevity phenomenon. And even more interesting, people found when other people living outside move into these areas, they also tend to gain this longevity magic. So clearly show that environment is very crucial to influence how well we age. But beside the environment, are there intrinsic genetic factors also making a contribution? So in the past few decades, advances in molecular genetics of aging now reveal many genes and signaling pathways that can regulate longevity. The first of such molecular mechanisms discovered was insulin IGF-1 signaling. Here is a very, very simple version of this pathway. So the insulin IGF-1 receptors is located on the cellular membrane, and upon ligand binding, it will be activated. Then through a series downstream phosphorylation event, the FOXL transcription factor will be phosphorylated, inhibited, and returned in the cytoplasm. However, when the upstream signaling component of this pathway are inactivated, now the FOXL transcription factor can translocate into the nucleus and activates target gene expression to promote longevity. So this longevity mechanism was first discovered in C. elegans, and is later shown to be well conserved in Drosophila and in mice. And more importantly, several centenarian cohort studies also confirmed the importance of this pathway for human longevity. So this discovery was very, very exciting for the community because at least tell us two things. So first, it showed there are genes and signaling pathways that can regulate longevity. And secondly, show that the molecular mechanism that we discovered in invertebrate can be translated for understanding human longevity. So since then, there are many, many more new genes have been identified. But then how do those genes exert their effect on organisms? So we know for any biological systems, cell is a functional unit. For a cell, genes are very important, and they hold the code that everything that could happen. But they are not the one directly doing the job. So following this genetic code, this information chain will be largely amplified through transcription and further translated into a variety of different proteins. And proteins will carry out most of the work in the cell, including driving essential metabolic reactions. During those reactions, various chemical intermediates and byproducts will be generated as metabolites. And because they are the endpoint of this information chain, so metabolites are most directly connected with cellular activities. And compared to other biomolecules, they are most conserved across different species. So in a way, they can be viewed as the most common currency used in biological systems from prokaryotes to eukaryotes. But at the same time, different from eukaryotic cells, eukaryotic cells have evolved a way to separate those metabolic reactions into different cellular compartments called cellular organelles. So within those cellular organelles, different metabolic pathways act there and generating unique pool of metabolite with different cellular functions. And this compartmentation is a very crucial component of metabolic regulation in eukaryotic cells. And it dramatically increases the specificity and efficiency, but at the same time generate a big challenge for the cell. Because for the cell to function properly, these different cellular organelles cannot just function completely independent from each other. They have to communicate to each other to coordinate their metabolic activities. But how do they do this? So in our research, we think metabolite will be excellent candidate as the signaling molecules for these intracellular communications. And those chemical dialogues between them are very crucial to maintain cellular homeostasis and ultimately organism fitness. So finding those messenger metabolites will provide us a new way to promote healthy aging. So in my lab, we aim to discover those messenger metabolites, to investigate how the metabolite-directed cellular communication systems works, and more importantly, to understand how can manipulate those systems to promote the healthy survival of organisms. And we address those questions in a free-living nematode species, Xenohybridis elegans. So these are multicellular animals with whole body transparency and a well-defined cell fate. So this offers us a unique opportunity to study the subcellular dynamics at the most physiological conditions in living organisms. Secondly, C. elegans are very powerful models for large-scale genetic studies, allowing systemic discovery of new regulatory genes and signaling pathways. Last but not least, our recent metabolomic studies demonstrate that metabolite profiles are well conserved between C. elegans and human. So therefore, we think the discovery of messenger metabolite and their regulatory network in C. elegans can be directly translated to other systems. So one of the areas we're recently focused on is a key cellular organelle called lysosomes. So these cellular organelles are loaded with a variety of different hydrolytic enzymes to break down lipid, proteins, carbohydrate, etc. And it acts downstream of endocytosis and phagocytosis to digest the materials taken up from the environment. It also interacts with autophagy and responsible for recycling all those damaged cellular components. Although long considered as the scavenger center of the cell, recent studies, including ours, indicate the signaling function of the lysosome. So in our work, we discovered a lysosomal lipase called LiPo4 in C. elegans. So in wild-type conditions, this lipase is expressed at very, very low levels, barely detectable. And we made a transgenic C. elegans to increase its levels and find animals live more than 50% longer than wild-type controls. So the two lines you see here in this graph called lifespan curves. So we follow hundreds of animals every day, record how many of them are still alive, and then plot the percentage of survival with time. So you can see for one type of animals, 50% of them are all dead around day 18. And at the same time, if you look at LiPo4 transgenic animals, 90% of them are still alive. And interestingly, these animals are not just long-lived, they are healthy long-lived. So worms are like humans. When they are getting older, their physical activities are dramatically declined. So here show you 18 days old wild-type animals comparable to people around age 80. So you can see these animals still moving their head and tail, but very inactive. In contrast, if you look at age-matched LiPo4 transgenic strains, at this age, they are still actively moving around on the plate, just like a young adult. So I think even for people who never saw C. elegans before, it's not very hard to tell these animals are much healthier. So now the question is, how does this lipase, the lysosomal lipase, make animals live longer and healthier? So chemically, we profiled over 300 metabolites that are conserved between human and C. elegans and found 4 lipid metabolites that are induced in the long-lived LiPo4 transgenic animals. And these include 3 C20 polyunsaturated fatty acid and 1 fatty acid derivative called oleic acenolamide. It turned out OEA is a very exciting target because when we give it to wild-type animals, now they live longer. And also these animals are more active when, in their later age. So this shows that supplementation of this natural compound can recapitulate all the beneficial effects we see in the lipase transgenic strains. And then next, further biochemical and genetic analysis also help us to review the regulatory mechanism of LEA. And so we discovered that upon the induction of the lipase in the lysosome, a fatty acid binding protein LBP8 will be translocated from the lysosomes and to the nucleus and function as a lipid chaperone to carry OEA together. Then in the nucleus, OEA will bind to its endogenous receptor NHR80 to turn on its target gene expression. And so together, these findings reveal the first lysosome to nuclear retrograde lipid messenger pathway, and then also identify a specific metabolite that can mediate the communication between the lysosome and the nucleus. And it showed the activation of this pathway will lead to organism longevity systemically. So this work really highlights the signaling function of the lysosome. In fact, in the cell, this organelle is in a perfect position to sense both environmental and intracellular stimuli. So in response to those input, various chemical reactions in this cellular organelle will generate a different metabolite. And we think some of those metabolites can function as signaling molecules to mediate the communication between other cellular organelles in the cells and even between cells. And we call this a lysokine hypothesis. So our work shows you one example, but I think we only revealed the tip of iceberg here. So we think there are many more lysokines waiting to be discovered, and they will be very exciting natural target for us to use and to promote healthy aging. So now you can see metabolites as a central component of my research. However, those molecules are very difficult to study. So recent years, advances in chemical analytic approaches, make their discovery easier and easier. But it's still very challenging to visualize those small molecules in living cells and organisms. So to study the spatial temporal dynamics of metabolites in living organisms, we have been collaborating with Sunny Hsieh's group at Harvard and applied stimulated Raman scattering microscopy into our research. So what is stimulated Raman scattering? It describes a phenomenon when two photons, pump and Stokes, interact with the chemical molecules. If their energy difference match to the vibrational energy of the chemical bond in these molecules, then through nonlinear interaction, it will stimulate the chemical bond to the vibrational excitation state. So as a result, some energy will be transferred from the pump photons to the molecules and then generate a new Stokes photon with less energy and longer wavelengths. But if their energy difference doesn't match anything, then nothing happens. Therefore, based on the characteristic chemical bond in the molecules, then SRS provide a very unique chemical contrast to image those molecules. So in the SRS microscope that we built in the lab, there are 2 picosecond pulse lasers, and one has a fixed wavelength and it's modulated at 10 megahertz high frequency. The other one is not modulated, but has a wavelength that you can tune to different range based on the molecule of interest. Then these 2 laser beams are spatially and temporally aligned and fit into a laser scanning microscope. After scanning the samples, if there is SRS effect, then the palm beam will lose photons and experience intensity loss in a modulated manner. Now the question is, how can we extract such a small intensity loss signal from the background? So what we do actually quite similar to what you do when you listen to a radio. So although there are very different radio signals in the air, you can always tune the radio to a specific frequency of your favorite radio station and listen to your favorite programs. So in our case, although the SRS loss signal is very small, it has a fixed frequency at 10 megahertz. So we can use electronic filters and the locking amplifier to extract this signal specifically and amplify it for image construction and quantification. So based on this 2-photon principle, SRS microscopy is capable of 3D imaging of chemical molecules at a diffraction-limited resolution in living organisms. So here show you one example. We use SRS microscopy to image fatty acid molecules stored in a specialized cellular organelles called lipid droplet and using symmetric carbon-hydrogen bond. So the left panel shows you 2-photon fluorescence microscopy image of paralipin A localized at the surface of a lipid droplet. And the middle panel shows you SRS imaging of fatty acid molecules stored in the core of a lipid droplet. So you can see the overlap as a subcellular resolution. So it's very exciting. First time we can see those molecules in vivo in living organisms. So using this new technology platform. So now we can image different aspects of lipid molecules and other metabolites in living cells, living organisms, and to reveal their interactions with proteins. And more importantly, it offers us a unique opportunity to investigate the molecular mechanisms regulating those dynamics and those interactions. So it's very exciting for us. So I will end here, and so thanks all the current and former members in my lab, especially Andy, who lead the first lysosome work, and also Yongyu, who is leading the microscopy chemical imaging project. And also want to thank all our collaborators from very diverse research areas. So I really appreciate they bring their unique expertise to our research and allow us to study the question we can never even imagine before. And also want to thank all the funding agency to support our work. And thank you for your attention. Yeah, so basically, it's in theory, you can use it for anything. But because it's true photon principle, the depths we can go, right now the limitation is about 100 microns. So that's why, like, C. elegans is perfect. We can image the whole organisms. But, for example, our collaborator who interested, like, brain or livers, then they have to section them to about, like, 100 to 200 micromolar, micrometer, like, thickness. Then we can image them. Yeah, but you can use it. Yeah, you can use it for any system because it's label-free. And then, so basically, you just cut them, then you can image them. Yeah.
You gave an example of two places, one in Italy and one in Greece, where the people live longer.
Yeah.
Did you make any study about when those people, say, immigrated to the United States or Canada, how long they live, that type of community people?
I don't think I don't think people did very serious scientific follow-up for those. But from the news I read, is some of them, they said they do lose this longevity magic. So they don't. That's why they don't think it's a genetic contribution, but they think it's environment. And actually, diet do play a very important role in this regulation because all those areas you can see they're next to this ocean. So their food are very rich, like in fish, for example, and olive oil. And also very interesting, a lot of those areas, they only have 2 meals every day instead of 3 meals. So, all those things people think make a big contribution to their longevity phenomenon. Yeah.
So it is the location.
Yeah, the location. I mean, I think that's the thing when you study environment, it's hard to dissect a lot of, very complicated and intertwined factors. Yeah.
Yeah, thank you.
Great presentation, wonderful.
Thank you.
The nuclear hormone receptor, I guess, the NHR. And you mentioned, obviously, maybe because of time constraints, you didn't mention the longevity genes that are upregulated or their expression is perturbed. Is longevity then governed by the epigenetic regulation of those longevity genes? Or is there a genetic hardwiring of those genes?
Yeah, so, first, epigenetic regulation do play very important role in longevity. Like my colleague Anne Burnett at Harvard, and they pioneered the work, that discover epigenetic factors is very important for longevity regulation in C. elegans. I think later on there are follow-up work in other organisms. But in terms of the genes that we study, whether or not epigenetic factors play a role there, it's still unclear to us. Yeah. So
which gene is that? Which genes are longevity genes?
For the epigenetic factors?
No, for all the genes that are regulated by NHR there, which are the key genes that govern the longevity of
Oh,
the
you mean
C.
the
elegans?
target of,
Yeah.
yeah, so from the transcriptional analysis, a lot of genes, actually there are two classes of them, and I don't have time to talk.
Sure.
So one class is the mitochondrial genes.
Okay,
So,
sure.
you know,
Makes sense.
it's very interesting then that showed there actually also a crosstalk between lysosome to mitochondria, you know, which is a cycling center. Another is like a basically it's an energy field of the cells. And so that's for all cell autonomous regulation. So I also didn't talk about this pathway specifically function in the fat storage cells. So it's kind of adipose tissue of the animals. So this is all cell autonomous regulation. But very interesting, a lot of the transcription target that we identify for the nuclear hormone receptors, a lot of them actually are secreting factors that affecting neuron functions. So actually we are more like recent work, we found there also a crosstalk between the fat and neurons. And then from neurons, they release the secondary messengers to coordinate the whole organism's activity. I think totally makes sense because longevity is a whole body phenomenon. So I think you also see this tissue-tissue crosstalk.
That's why maybe less depression.
So can I ask you one last
Yeah.
question? Does that mean that your mother's is more important in terms of aging if half of the genes come from the mitochondria? Is that?
That's interesting. Let me think how actually, I don't have a short answer for that because I don't think I see any study on that, but it's an interesting hypothesis. Yeah.
Thank you.